Cancer stem cells are implicated in cancer relapse, metastasis, and drug resistance. This protocol provides an efficient method for investigating the functions of KN genes in cancer stem cells. Using a conditional lockdown system and adapted cell formation assay, this method is easily adapted to study in vitro and in vivo functions of Stemness-Associated genes in different types of cancer stem cells. Demonstrating the procedure will be Yuting Li and Xiangyu, research assistants from my laboratory.
To generate lentivirus particles, seed 4,293T lentiviral packaging cells into a 100 mm Petri dish with 10 ml of DMEM supplemented with 10% FBS. Incubate the cells overnight at 37 degrees Celsius and 5% carbon dioxide, making sure that the cells are 50 to 80% confluent on the day of transfection. Bring reduced serum medium to room temperature and prepare tubes A and B according to manuscript directions. Add the contents of tube A to tube B. Mix well and incubate the tube for 15 minutes at room temperature to prepare lipid DNA complexes. Remove 5 ml of medium from the cell culture dish. Then add 5 ml of the lipid DNA complex